Students, our today's topic is Auxin Structure, types, and biosynthesis. So, it is the first plant growth hormone to be studied in detail. It plays important role in growth and development of plants like stem elongation, apical dominance, root initiation, fruit development, meristem development, everything. Um, when we observe organogenesis in the first zygotic cell, this is all that was also controlled by auxin. The term auxin is derived from the Greek word auxein, that means to grow. So there is naturally occurring auxin as well as synthetic auxins. Auxins passes from the shoot tip to the region of elongation, and its movement is basipetal in case of stem and acropetal in case of roots. So auxin helps in elongation of both shoots and roots. Auxin biosynthesis occurs in shoot apices, leaf primordial, and developing seeds from the amino acid tryptophan aromatic amino acid one, and with the presence of zinc ion. So the naturally occurring auxin is the indole acetic acid. Very simple structure. You can remember it easily. So try to remember and practice the structure. So these are some synthetic auxins like 2,4-dichlorophenoxyacetic acid. There are other synthetic auxins as well like naphthoxyacetic acid, 2,4-T, etc. So there are multiple uses of these synthetic auxins also. So they are widely used in agriculture and horticulture. I think you have heard about Agent Orange. This is a mixture of synthetic auxin 2,4-D and 2,4,5-T, which was used in Vietnam War to defoliate the trees of Vietnam jungles. Now please come to the biosynthesis part. IA biosynthesis is associated with rapidly dividing and growing tissues. Primary site of auxin synthesis are shoot apical meristem and young leaves. Root apical meristem also synthesize auxin. Young fruits and seeds also have high level of auxin. So its biosynthesis has two different orientation. One is tryptophan dependent process, another one is tryptophan independent process. But today we are discussing about only the tryptophan dependent pathways. There are four different pathways which are tryptophan dependent biosynthetic pathway of auxin. That is the IPA pathway, indole 3 pyruvic acid pathway, the TAM pathway, that is the tryptamine pathway, the IAN pathway, that is the indole 3 acetonitrile pathway, and the IAM pathway, which is the indole 3 acetamide pathway. So please come to the detail. Okay, so first of all, I am explaining the IPA pathway. So this is tryptophan, hmm, the precursor of auxin. From tryptophan, tryptophan transaminase enzyme produces indole 3 pyruvic acid. Then IPA decarboxylase produces indole 3 acetaldehyde, and from that compound, finally indole 3 acetic acid is produced. This is the IP pathway. In TAM pathway, TRP decarboxylase enzyme converts tryptophan into TAM, that is the tryptamine. Then amine oxidase enzyme converts it into indole 3 acetaldehyde, and from indole 3 acetaldehyde, dehydrogenase enzyme produces indole 3 acetic acid. In IAN pathway, from tryptophan, indole 3 acetaldoxine, a special compound is generated which which is again converted into indole 3 acetonitrile. From IAN, nitrilase enzyme produces indole 3 acetic acid. There is another pathway, that is the bacterial pathway. Here, from tryptophan, indole 3 acetamide is produced, and IAM hydrolase enzyme converts IAM into indole 3 acetic acid, or IAA. So, students, please go through the structures, try to remember all the steps and enzyme names, and happy reading.